Everybody, it's my honor today. For those who don't know me, I'm Dr. Chen, Dr. Casey Chen. I'm a physics professor here. It's my honor today to introduce to you Dr. Zhongrui Li. He's a friend of mine, good friend of mine, and I'm so happy to have him today. He is currently a senior scientist at Florida State University's Center for Advanced Material, High Performance Material.
Yeah.
Did I get it right?
Yeah,
okay.
Exactly.
Dr. Li got his doctoral degree from the chemical physics department. So he's a chemical physicist, actually. From the University of Science and Technology of China in 2000. The University of Science and Technology is one of the top universities in China. Actually, probably the best science and technology university. And after he finished his doctoral degree, he became a research scientist at a number of institutions. For example, Free University of Berlin and the University of Georgia, our flagship university here in Georgia, and University of Oklahoma, University of Arkansas, before he joined Florida State. My understanding is, in the past year, he did quite a bit of work in nanotechnology and development of nanomaterial, for instance, solar cell material, carbon nanotube. And as a result, Dr. Li has 5 patents under his belt related to this kind of technology. He can tell you more about it if you want to know more. Okay. Dr. Li's research is specialized in catalysis, nanotechnology, and synchrotron radiation. Currently, his interest is in developing nanomaterial for energy and biomedical applications, and that's why this is his talk today about applying nanotechnology, nanomaterial to Thermolysis of Cancer Cells. Let's welcome Dr.
Thanks for invitation and introduction. As I know, the Albany State University is very strong in biology and in biomedical forensic science. You can tell even the chair of the Department of Natural Science is a biologist.
I'm sorry, I forgot to mention one thing about Dr. Li. Dr. Li has published over 100 scientific papers in high caliber scientific journals.
Okay. I have done some research in bio-nanotechnology. We do have some overlap here. I'm glad to take this opportunity to present some findings in the nanomaterials for cancer therapy. First, I will talk the challenge of solid tumor treatment and the role of nanomaterial and nanotechnology in nanomedicine. Then I give the first example, use magnetic nanoparticles to small ablation of cancer cells. Then I talk the second project on the fluorescent magnetic nanocomposite for the radio frequency small destruction of cancer cells and also for imaging. Then come to conclusion and the final perspective. What is cancer? Cancer is a big disease that kills a lot of people. Cancer usually is caused by uncontrolled division of abnormal cells in a part of the body. There are 3 features that is a challenge we need to solve before we can treat the cancer cells. First, the cancer comes from our body, so it's a domestic terrorist. We know that bacteria and virus can be spot, but cancer cells are difficult to detect and difficult to treat. Cancer are also different from patient to patient. It's, each tumor is like a salad in the salad bar, having a different gradient. I mean the DNA error, because the cancer usually causes the DNA error. Cancer also continually change during the growth. Tumors are already playing the lottery, try to get the combination to spread themselves. In biology, we call it metastasize. These are many three challenges that nanotechnology need to be solved. What is nanotechnology? Nanotechnology is a creation or manipulation of the materials and device with at least one dimension sized from 1 nanometer to 100 nanometers. So it's very tiny as compared with the cells. Cells usually in the micrometer scale. So the nanomaterials and the nano device are very tiny, has a very unique properties and functions. They can enter into cells. I can use for the cancer cell the detection and the treatment. But we also know that cancer is very complicated. So in order to successfully treat cancer cells, multidiscipline approach are needed. This includes the genetic biology, material science, chemistry, and physics. Nanomaterials such as like quantum dots, magnetic nanomaterials, and carbon nanotube, gold nanoparticles, they all have a very unique optical, electrical, even magnetic properties. Nanotechnology can be used for the early cancer detection and treatment by combining using these advanced nanosystems with targeted delivery of drugs and a gene at a single cell level. So nanotechnology brings us hope to cure cancers. Nanomaterial can be used in many ways in the cancer treatment, such as drug delivery, imaging, and also the anti-drug delivery and early cancer detection. My research focuses this part, use nanomaterials to cure cancers through heating. Magnetic nanomaterials is a very good heating agent, can be used to localize heated cancer cells under radio frequency excitation. We know that nanoparticle is very tiny. It's also very active chemically. So it's unstable in air. So we need to coat it with something to protect it in the metallic state, so to prevent oxidation. So the magnetic particle can have a higher magnetic moment than the oxides. They also can, once it's coated with something, it also can increase the thermal stability and enhance the biocompatibility. Also can lower the toxicity. The outside coating also makes the functionalization possible. They maybe can use for special, as the targeted delivery. Here we use RF heating. Why we use RF heating? RF induction heating has some advantages than other source. RF, has a low frequency, can penetrate the body deep. So in principle, RF heating can kill all cancer cells at any place in the body. But we need the good thermal agent. Here we use magnetic nanoparticles. How to make this kind of coated magnetic particles? From carbon nanotube synthesis, we got some hint. This is a schematic diagram how carbon nanotube grows from metal particles. Usually, these metal particles are iron, cobalt, or nickel. This kind of transition metals. We know that actually carbon-containing molecules decompose on the cut its surface. They arrange themselves, then form a carbon-covered layer. When more carbon atoms added into this system, that system became unstable. That grows the carbon nanotubes. If we stop in the middle, so before the nanotube grows out, we get the coated, carbon-coated nanoparticles. So this is our method to grow to synthesize carbon-coated magnetic nanoparticles. Usually, grow carbon nanotube takes like 1/2 hour to 1 hour, but here we only let the reaction happen for 5 minutes. So after purification, we should get the gravity shield-coated nanoparticle. Let's see. This is the atomic force microscope and the TEM images. It's the AFM. You can see the particle, this dot. First image is a carbon-coated cobalt particle. You can see this lot of particles are coated with carbons shell. From the TM image, we can tell the cobalt particle is a very size around 2 nanometer. This is iron particle. It's 8 nanometer. The iron cobalt alloy is around 9 nanometers. Okay, they're all very protected by the graphitic layer. Sure. The layer is about 2 to 7 layers of a graphitic material, carbon layers. Okay, how do you know this particle is in metallic state, not get oxides during your synthesis or treatment, later post-treatment? We use the X-ray photon-electron spectroscopy to check this binding energy of the each element. Because each chemical state has their own specific binding energy. So we check this cobalt and iron. So to see that this very much very well with metallic state. If the cobalt is oxidized, so we should see the satellite peak like 5 to 6 eV above this 2p 3/2 peak, main peak. But we didn't see it here. So it means the cobalt is very protected and in the metallic state. This is the close look at the TM image. This is a cartoon. Okay, now we have a very good carbon-coated magnetic nanoparticles. How to kill the cancer cells? We use RF system. This is a device system instrument. They can generate RF frequency at around 350 kilohertz. We put the HeLa cancer cell. HeLa cervical cancer cells is usually called a normalized cell. These kind of cells can produce themselves as long as the survival conditions are met. So this is very commonly used for the biological, for cancer research. Right? Once you turn on the RF generator, so this will generate current inside this coil. Then that's oscillated in the magnetic and electric field. So that's on the magnetic particles inside will generate heat. Okay. So another thing I want to mention here, we use a very low frequency, 350 kilohertz, much lower than the current clinical microwave radiometer. They use a gigahertz frequency. We know the electromagnetic field penetration in the human tissue, human body. The penetration depth is inversely proportional to the frequency. Use this such low frequency, the electromagnetic wave can penetrate 15 centimeters of the tissue with more than 99%. I mean the intensity almost no change. So it's very efficient. That's why I said use RF heating can kill the cells inside any of body. Okay, let's see how the effect use the RF heating, local heating. Let's see this. Here we choose a very low concentration. 0.83 microgram per milliliter is very low, very safe. I mean the safe is biocompatible, not toxic. So you can see these curves. All this increase the death rate increase with the heating time. But we can see this iron nanoparticle has highest kill efficiency than other material like iron cobalt alloy and the cobalt nanoparticles. Now, if we keep the IV heating time as constant, let's say that 2 minutes, we found that the death rate of HeLa cells increased with the concentration of nanoparticles in the media. Especially the iron nanoparticle has a high killing efficiency. With almost 3.5 microgram per milliliter, the iron nanoparticle can kill almost 100% the HeLa cells. For the cobalt nanoparticles, it's red, much high concentration, like 20 microliter per milliliter are required to get the same therapy effect as the iron nanoparticles. Okay, how the nanoparticle change, temperature change under the RF the heating. Here is I have time. You can see this 5 minutes is enough to highlight the heating rate and the induction temperature of these nanoparticles. This blue light is an iron nanoparticle. It's much more efficient. They can reach almost 100 Celsius. It's 93 something Celsius. Right? The temperature also we found increase with the nanoparticle amount. The more the material, the temperature is higher. So among the 3 different types of nanoparticles, iron nanoparticles can reach even higher temperature. Okay. What's the reason? Because the iron nanoparticles is most efficient in the heating. We need to look at the mechanism. There are several ways, several processes happened during the magnetic reversal that generate heat. First one is called hysteresis loss, just like electromagnetic wave used in the home for cooking. If you have a ferromagnetic material, press AC current, they will reverse the magnetic field. Each time they lost some energy, that's as a heat. Right? The heat generator is proportional to the frequency it used, and also is proportional to the magnetic field, the strength you applied to the ferromagnetic material. The other source is called the Neal and the Brown relaxation. As the particle, magnetic particle, when the particle size shrink to the nanoscale, okay, they will change. That's the energy barrier for the magnetic reversal will decrease too. Okay. Consequently, this energy is even lower than the thermal energy, like a temperature. They will flip randomly. Okay, you do not need to apply the magnetic field. It flips on magnetic automatically, randomly. Okay. This is called Near relaxation. This can also generate heat. Okay. This momentum change inside the magnetic particle. If the magnetic particle itself can rotate inside the media on the electromagnetic field, this is called Brownian relaxation. Brownian relaxation also can generate heat. That's through the friction. The friction between the rotating nanoparticles and the medium, and that kind of called the friction loss in the viscous environment. And also we call it the eddy current. Eddy current is also due to the alternating electromagnetic field in the conducting material. We know the iron cobalt alloys, they are metallic, so they can also generate eddy current. But keep in mind, this nanoparticle is very tiny. Our nanoparticles is mostly below 10 nanometer, so the eddy current is negligibly small. So compared to these other 3 sources, this contribution to the heat generation can be neglected. Okay, Let's do some magnetic property measurement for our nanoparticles. We measured the magnetization curve at the very low temperature, 5K. It's room temperature, 300 Kelvin. We can see that our nanoparticle has a very high hysteresis loop, large area. We just said that a large hysteresis loop can generate more heat when during the magnetization. Okay? At the room temperature, this loop, the shrink is purely for cobalt nanoparticles. Almost nothing there. But they still have a higher saturation magnetization. This kind of phenomenon is referred to the superparamagnetic. When the particle size shrink, okay, that goes through this single domain. Even the smaller than single domain, it will enter into this zone. This is called superparamagnetism. Just as I mentioned, superparamagnetic nanoparticles, the magnetic moment inside can flip randomly, even on the heat, on the room temperature. Okay. So just like a ferromagnetic material have a Curie temperature, okay, superparamagnetic nanoparticles has a called blocking temperature. What is blocking temperature? Blocking temperature means below this temperature, the magnetic moment are frozen, like blocked, just like treating that as like a magnet. You cannot change at the low magnetic field. You cannot make any change. But above this is just like a paramagnetic nanoparticle. Even it's below the Curie temperature. Usually the ferromagnetic material below the Curie temperature is magnetic. Okay, it's different. How to get the blocking temperature? We need to do magnetization on the different cooling technique. We always can get two type curves. One is called the field cooling. It means from room temperature to the very low temperature. Here is the helium temperature, the 5K. In this case, the material cooling on the magnetic field. This is called field cooling. Another is called zero field cooling. The cooling without a magnetic field. Okay. For the FC, this is called a magnetic. Field cooling. The curve they all decrease with the temperature. A magnetic moment decrease with temperature. But if you look at the zero field cooling, first they increase with temperature, then left off. This is like a crossover temperature we call the blocking temperature. For the superparamagnetic nanoparticles, the heating effect directly associates the blocking temperature. Usually the higher the better. Higher the blocking temperature, the higher the heating generation efficiency. So you can see this is cobalt. That's like around 50 Kelvin for the cobalt nanoparticle. The iron cobalt alloy is around 250. For the iron nanoparticle, the blocking temperature is 270 to 280, so much higher than that. This will give you the answer how the iron nanoparticle is the most efficient in the cancer killing on IF excitation. Okay, let's see what happens to the HeLa cells. Once first image is we got from the confocal optical microscope. This is just the HeLa cells, but this is without the incubation of a magnetic particle. This is after 2 days incubation. That's the magnetic cobalt nanoparticles. You can see the aggregated inside the cells. You can also even find some nanoparticles inside the nucleus of the HeLa cell. It means the nanoparticle is very tiny, 10 nanometer. They can go through into the cell, even can cross the nuclear membrane into the nucleus. The image you see is a membrane blabbing of the HeLa cell incubated with carbon-coated iron particle, just for 2 minutes. You see what happened. Okay. Then image D, E, F, we compare 3 different nanoparticles to see what the killing effect of these nanoparticles. Okay, this is cobalt nanoparticles, this is alloy nanoparticles, this is iron. The green cells dyed with green, it means it's still alive. It's the orange or even the browning or something this kind of color means the cell is already dead. Okay. So apparently iron nanoparticle is the best among these three different nanoparticles. We also see some difference. Why is some cells are still alive, some are not? That's because nanoparticle has entered into this, more nanoparticles enter this cell, so this causes the die. So this gives some information. So nanoparticle kills the cancer cells not due to the particle raises the whole cell temperature to very high temperature. Actually, this is because due to the localized heating. Only the nanoparticle, the side of nanoparticle, the temperature can raise to very high. Usually can raise to the 90 Celsius. This is higher than the temperature the cancer cell can stand. Okay, so this is the RF heating. This typing wrong. Magnetic nanoparticle causes this localized heating because the heating can destroy the membrane, can make the DNA the fragmentation, and also the thermal denature of proteins. Protein can only stand below up to 50 Celsius. Right. We also did the electrophoresis. This you can see that this standard like a mark DNA. You can different like zebra lines. Most of you have already saw this. Okay. The second lane is the DNA extracted from HeLa cell without any nanoparticles and no RF exposure. So the protein DNA the aggregated in this range here. There's no diffusion. Okay. The third one is the DNA of the HeLa cells incubated with a single nanotube and exposed to RF. You already see some of the change here, the color change. Okay. Last one, the fourth column is the DNA of HeLa cell incubated with carbon-coated cobalt nanoparticles and exposed to the RF. So you can see something here. It's a gray light. So it means there's some DNA fragment located in this range. So this indicates that DNA is fragmented and generates small fractions in the HeLa cell from HeLa cell that's incubated with nanoparticle on the RF excitation. Okay. So next topic I want to talk about how to use quantum dots for the imaging. Imaging is important because you especially want to study the cancer therapy and some other biology process. The basic idea here is to combine the iron oxide nanoparticle with quantum dots. But the iron nanoparticle cannot directly connect to the quantum dot to make a composite. We need to make a change to modify the surface property of the iron oxide particle and as well as the quantum dots. How to do that? First, we coated the iron 304, the iron oxide. We also call it magnetite, this kind of nanoparticle with the silica through the gel process. We did the XRD to check this the crystal structure. This matches the magnetite very well. So we synthesize the magnetite nanoparticles. This is a TM image of the magnetite nanoparticles. You can see this size is around like 10 nanometer. This is the composite. Actually, is iron oxide coated with silicon oxide. This shell you can see is around 4 nanometers. Okay, we make one part. This is a magnetic particle. We still need to make to modify another quantum dots. The quantum dots still need to be modified. Here we use cadmium selenide. Cadmium selenide itself not toxic, but in the biology environment, it can release cadmium ions that are toxic. So we have to make some treatment. So we coat it with zinc sulfide. This coating can improve the stability of this nanoparticle in the biology environment. So another thing is no significant surface absorption will happen on the surface, surface absorption of a biomolecule onto this particle. This also has an advantage for the quantum efficiency of the quantum dots, because the quantum dots, if quantum dot surface is covered by the biology molecules, the quantum efficiency will decrease. That's called quantum quench. So we still can get a high quantum efficiency. Okay, and the third thing I just mentioned, this kind of coating can lower the toxicity of the quantum dots. This is the quantum dots on the UV irradiation. We see the irradiation, the light is this is a red light. Wavelength is around 600 nanometer. Right side image is a TM. You can see this is the quantum dots coated with zinc sulfide. We also did the optical absorption for this material. This is water-soluble quantum dots. Because this coated this zinc side layer, it became water-soluble. It's very good for the biology research. This red curve is iron oxide, no much absorption. Here is coated quantum dots. You still see it here. We also did the index to see the checks all elements, because after synthesis we wash this with acid or water, to see if all these elements are still there. Okay, we found this exactly all these combined together. See that? Okay, this is a TM image. You see the black dots hide. These are the iron nanoparticles. The small dots around the dark dots. I mean, this looks pretty great. This is from the cadmium selenide quantum dots. Right. This is the schematic diagram of our composite. So from the image, we can see the average quantum dot is about 5 nanometer. This coating shell is 1 nanometer, very thin. Okay. How this coating treatment affects the magnetic property? Is that still magnetic? We did magnetization for all these materials. The black curve is iron oxide. You see, have a very strong magnetization value. After coated with silica, this magnetization drop. It makes sense because silica is a non-magnetic material that is screening the magnetic field. So that's magnetization drops. When first coated joined with quantum dots, the magnetization value drops again, but it still has a lot. Okay. So to further verify the magnetic property of this composite we just made, we can use a magnet. Put on side of this bottle solution. Usually it's a Brownian solution, very uniform. But put a magnet, you can separate the nanoparticle, the nanocomposite, from the chloroform solution. Right? So nanocomposite will synthesize as your magnetic. Right? So now we use this nanocomposite to treat the pancreatic cancer cell. The former CEO of Apple company, the Steve Jobs, died of this kind of cancer cell, the pancreatic cancer cell. Right? Let's see the different concentration was added into the medium during the incubation of the pancreatic cells. Without the nanoparticle, just turn on the IF heating for the 2 minutes. Doesn't kill much of the pancreatic cells. But when you increase the nanocomposite, you can see that's death rate, death percentage of the pancreatic cells increase with concentration. We found that if concentration above 1.66 microgram per milliliter, almost all pancreatic cells can be killed just within 2 minutes of RF excitation. Right. The figure B is the confocal microscopic image of the normal pancreatic cells. The C image is nanoparticles that incubated with the pancreatic cell. That's distributed pretty uniform generally. You can see the bright dots. That's due to the quantum dots, because this nano composites are under excitation of UV, ultraviolet excitation. They generate red light. Right. The image D is we got from the nanoparticles under excitation, UV excitation. That's what I'm taken into the cell cytoplasm of the pancreatic cells. You can see some particles go into this is the membrane of the cell, and also go inside, even can go to the nucleus. So it means this particle is just still small enough, it can diffuse into the cell and even to the nucleus through different channels. Okay. That's the image E to this process. Once we turn on the IF at a different time, we take the image. First one you can see this apparently shrink. This is about cell, the pancreatic cell's diameter is around 20 micron. Here is just like a 10 micron. It's a shrink. First the shrink. Okay. Then you can see some disintegration of nuclear membrane happened in this. This uses kind of nuclear, but here you could not, because the nuclear is membrane is disintegrated. Then this image G, this you can see that's almost all disintegrated of nuclear membranes. This it's not like a cell anymore. So last is image is apoptotic body of the pancreatic cells. This is called thermolysis. Right? It's good. We killed all the pancreatic cells with a low concentration of nanocomposite in just a short time. Okay. Now to come to the summary, and we successfully developed the two types of nanoparticles. They're all magnetic. First, use carbon-coated nanoparticles, iron cobalt alloy. We found the iron nanoparticles more efficient, can kill the HeLa cervical cancer cells. Second is material is we combined the magnetic particle with quantum dots, so they have imaging capability. So all these particles are in nanometer size, usually below the 10 nanometer. They can penetrate the cell membrane and even go to the nucleus of cells. So we use RF heating. This is very efficient, can penetrate the whole body tissue. So this can treat any kind of cell, any place inside the body. So this technique is very useful, especially in the vivo biomedical treatment. More importantly, this kind of material can be manipulated. Also can be tracked, because we added like quantum dots can be used for imaging. This is the RF heating that can use remote heating. You do not need to put heat inside the body, so you just put the RF generator outside the body. It can kill the cells inside the tissue. Okay, this is a general process of how to use magnetic nanoparticle kill the cells. First, we incubate the normal cancer cells, then culture these cells with nanoparticles and turn on the RF heating. So these nanoparticles, destroy the membrane and destroy the DNA, destroy the nucleus. Okay, so you got the cell lysis. The cell is the cancer cells killed. Okay, so now have developed a very good bomb to kill the domestic terrorist, but we still need a vehicle how to deliver this bomb to the specific the cancer cells or cancer tumor. So we need some specific. This is pretty hot and also challenging field in the cancer research. Some people used to try to coat the nanoparticles just as a light synthesized magnetic particle with some ligand. These special ligands can with touch of some specific receptor inside on the tumor cells. A lot of people are doing this kind of research. That is also reason attract a lot of attention in this field. Nobel Prize committee award 3 scientists, American scientists doing this field. They developed discovered the machinery that regulate how cell transport major molecules in a cargo system. So we need a specific cargo system to develop nanoparticles to the specific cancer cells. That's the reason to the right place at the right time that they got the Nobel Prize of this year. That was announced tomorrow, or yesterday, yeah, Monday. Okay, so that's my talk. Thank you.